Hello everyone, my name is Arpita Routray and in today's video I will be explaining the topic of regulation of gene expression in prokaryotes. Okay, we will understand this process by giving a special emphasis on the model of Lac Operon. Okay, we will understand Lac Operon properly. So let's begin. Before going into gene expression thing, we first know where the gene expression is taking place. Gene expression. Okay, and what we mean by the process of gene expression. Okay, let's understand. We all have heard about the term central dogma. What central dogma says? Central dogma says the flow of information from the DNA to RNA, and from RNA, the information is transferred into protein. Our body or any organism or any living organism is having thousands and thousands of proteins in its body. Thousands of molecules are present, thousands of functions are occurring. So these functions need to be regulated. And the information for this function to occur or not to occur is present where? It is present in the DNA. We all know that all the information of our body is present in DNA. Now, this DNA, the information which is present in DNA, it needs to be converted into what? It needs to be converted into a protein. And this process occurs in two steps. What we know, first step is our transcription, and the second step is our translation. What happens in transcription? The information present in DNA, coded information present in DNA, is transferred into an RNA in coded form, and now this code present in RNA is again transferred into protein. So this thing takes place. So now let's understand how it happens. So when we are talking about gene expression, what we mean? We exclusively mean the process of transcription. Why transcription? Genes are present in where? Genes are present in DNA. So when we are talking about gene expression, we mean the formation of RNA from the DNA. Okay, it is clear now. Now let's move to operon. So what operon is? Before going into the points of the slides, I would like to explain the operon concept by here, giving a simple example here. The DNA, suppose this is a prokaryotic DNA. Okay, suppose this is a long fragment of a prokaryotic DNA, long DNA of a prokaryote. Now, this DNA is having several genes. Suppose 1, 2, 3. I'm giving here examples of 3 genes. Suppose this is our gene A. Second, this is suppose gene B. Now, third is suppose gene C. Likewise, several genes are present, gene D, E, F. Just for example, I am giving the names A, B, C, D, whatever. So, these genes are present where? In the DNA. Now, what happens in prokaryotes? All the genes are not switched on at the same time. They are only switched on when they are needed. Suppose the body needs gene A, the product of gene A. Then what the prokaryote will do? It will switch on only gene A, not B, not C. Okay, according to the need, it switches on and switches off. So what operon is basically, where this operon is situated in this fragment of DNA? Operon is basically these genes which are present, okay, these genes and certain regulatory elements. These genes are accompanied by certain regulatory elements. Suppose this is a regulatory elements. Okay, likewise for gene B, there are certain regulatory elements like this. Gene C, there are certain regulatory elements. So what is an operon? Operon is what? Operon is a unit of bacterial gene expression and regulation. Unit of bacterial gene expression means what? It is having the gene and the regulatory proteins. Okay, regulatory fragments. Okay, these whole thing constitutes an operon. Operon. I hope the concept of operon is clear. Now, let's move on to the components of operon. First, who described the concept of operon? Jacob and Monod in 1961, they first described the concept of operon. Now, moving on to the components of operon. The operon consists of 4 components. 4 components. What are the 4 components? Number 1 is our regulatory gene. Second is a promoter gene. Third is an operator. And fourth are the structural genes. So what are the functions of these gene fragments which are present in the DNA or which are present in the operon? First one is regulatory gene. What regulatory gene does? It regulates, as the name suggests, it regulates the process of switching on and switching off of the operon. How it regulates? It synthesizes a repressor protein. What it does? It synthesizes a repressor protein which regulates the process of switching on and off. And how the repressor protein regulates? This repressor protein, suppose this is a repressor protein which the regulatory gene is synthesizing. This repressor protein or repressor RNA goes and binds with the operator region, and hence it regulates. So now we have studied the function of two components of operon. Number one is regulator. What regulator is doing? Regulator is synthesizing a repressor protein. Number two is our operator. What operator is doing? Operator is providing the binding site for the repressor protein to bind. Okay, operator is providing the binding site. Number second is our promoter. What is the function of promoter now? Okay, promoter, what happens? How the DNA, when the transcription process takes place, suppose this is DNA, okay, DNA to our RNA is needed to be synthesized. Who does this? This is done by an enzyme which is called RNA polymerase. RNA polymerase. What RNA polymerase does? RNA polymerase simply moves over the fragment of DNA like this. It simply moves over the fragment of DNA, and along with its movement, it will simultaneously synthesize an mRNA, messenger RNA or any RNA. It will synthesize an RNA. So this way, what happens? An RNA is synthesized. So this movement is restricted. Movement is either restricted or allowed by the promoter itself. Promoter, as the name suggests, it will promote. It will promote the movement of what? It will promote the movement of RNA polymerase. So RNA polymerase, this enzyme will bind here, and promoter depends. Promoter defines whether this will move further or not. So what is promoter? Promoter is the binding site for RNA polymerase. Three components and its functions we have studied now. Now the last component is our structural gene. What are structural gene? These are the basic genes which have the coded information for the protein which we want, protein of interest, therefore we are calling it protein of interest. So these are the gene 1, gene 2. These whole thing is the structural gene. Okay, the structural genes are having several small genes, gene 1, 2, 3. It depends. There can be 3, there can be 4, there can be 5. We will see in the further examples. So these structural genes are having the protein of interest. The protein which we want to be expressed are present where? It is present in the structural genes. I hope the concept of operon is clear now. Now let's move on to the types of operons which are present. Okay, basically two types of operons are present. What are the two types of operon present? Number one, an inducible operon. Number two is a repressible operon. What is an inducible operon? As the name suggests, induce means they are induced to do something. This operon, the type of operon which is usually off, okay, which is very lazy, I must say, which is very lazy and which is usually off, and it is switched on by a molecule, and that molecule is called inducer. Okay, inducer molecule, what it does, it induces the off model of operon to get switched on. Okay, this whole process is done by the inducer, and since the inducer is inducing the operon to switch on, therefore it is called an inducible operon. Okay, example of such operon is our Lac Operon, which we will study today. Second one is a repressible operon. What happens? The reverse case occurs in repressible operon. That is, the operon is usually is in on condition. That is, the protein which is to be synthesized, it goes on synthesizing continuously, which is always in an on position, and it is needed to be turned off. It is needed to be turned off. It is needed to be what? It is needed to be turned off. And this turning off process is done by a co-repressor molecule. So when a co-repressor molecule is added, what happens? The switched on operon is turned into switched off position. So these are the two types of operon. Number one is an inducible operon, which is usually off off in position, and it is turned on with the help of an inducer. And second one is a repressible operon, means which is an usually on condition, and it is repressed with the help of a co-repressor. Okay, and it is switched off. Now let's move on to the example of Lac Operon. So this is Lac Operon. What is a Lac Operon? Just right now we studied Lac Operon is the inducible operon. Okay, and what for Lac Operon is used? The function of Lac Operon, it is used for the lactose metabolism. Lactose metabolism. Metabolism means lactose metabolism. What does metabolism means? Metabolism means it can be either anabolism, that is synthesis, or it can be catabolism, that is breakdown. Okay, so lactose for Lac Operon is used for the purpose of lactose metabolism. Okay, what happens when suppose we have certain bacteria here. Okay, there are certain bacteria here, and the bacteria needs food to grow, and the food of bacteria is nothing but our lactose. Okay, but the bacteria is unable to utilize this lactose. For being able to utilize this lactose, certain genes are present in its DNA which are needed to be switched on. Okay, and this switching on process of the genes for utilization of lactose are present where? Are present in the Lac Operon. Okay, so here what is the inducer molecule? Here the inducer molecule is lactose. Whenever lactose will be present in the medium, it will induce the DNA of the bacteria, of the prokaryotes, to synthesize, to what? To switch on what? To switch on the Lac Operon so that the lactose can be utilized. Now let's see the components. Here the same thing is written which I have said right now. What happens? So Lac Operon is an inducible operon that encodes enzymes for the metabolism of the sugar lactose. Okay, it turns on only when the sugar lactose is present, and other preferred sugars are absent. The inducer here is an allolactose. The lactose and allolactose, they are almost the same thing, but allolactose is the more suitable term. That is a modified form of lactose. As I said, both are the same thing, but it is a suitable thing. Now, moving on to the components of Lac Operon. As we studied the general components of an operon, similarly, the components of Lac Operon are also same, but we need to remember the three structural genes which are present. Here, we will understand it with the help of this diagram. These are the structure of Lac Operon. It is made up of one regulator here, second is a promoter, third operator, and fourth is a structural gene. These are present. Now this structural gene is again made up of three components. We will see what are the three components. The three components or the three genes are Z gene, Y gene, and A gene. These Z, Y, and A have different functions. What they do? The Z gene is responsible for coding beta galactosidase. Beta galactosidase. Y gene encodes what? It encodes permease, and A gene encodes trans-acetylase. Okay, trans-acetylase. So these are the different genes, and these are the different enzymes which are synthesized by the respective genes. Z genes for beta galactosidase, Y gene for permease, A gene for trans-acetylase. Okay, let's see the function of these enzymes. Beta galactosidase, what is the function? It catalyzes the hydrolysis of lactose into glucose and galactose. Okay, lactose is a molecule which is needed to be uptaken by what? The bacteria. So for suitability, what the bacteria does, it synthesizes an enzyme which is beta galactosidase. Now suppose this is a lactose molecule, and lactose molecule is made up of two components. One is glucose, second one is galactose. What this enzyme does, beta galactosidase, what does? It does the hydrolysis of this enzyme, hydrolysis of lactose, sorry. It performs the hydrolysis of lactose and separate this one was the whole lactose molecule, and it separates into two components. One is glucose, second one is galactose. Second one is galactose. So this is now a simpler form and easily digestible form, right? So that is why beta galactosidase hydrolyzes the lactose molecule. Second gene is our Y gene. What is the function of Y gene? Y gene encodes permease. Okay, what permease does? Which regulates the lactose permeability to the cell. It regulates the lactose permeability to the cell. What happens? Suppose this is a cell. Now we have what lactose is broken down into glucose, and second component is our galactose. Now these glucose and galactose need to be permeabilized into the membrane, into the cell membrane of the bacterial cell. Suppose this is the bacterial cell. Okay, and this permeability is enhanced by the enzyme permease. Permease allows the glucose or galactose or the whole lactose molecule to get incorporated into the cell. This is the function of our Y gene protein, that is the permease. Third gene is our A gene. A gene encodes transacetylase. Transacetylase. What is the function of transacetylase? Transacetylase is just a helper, and what it does is assist the enzyme beta galactosidase. Beta galactosidase does the breakdown of lactose molecule into glucose and galactose, and its assistant is our A gene. Now let's see the components here again. Okay, this is the structure of Lac Operon, which is made up of regulator, then promoter, then operator, then structural gene. Structural gene is again made up of three components. These three components are our lacZ, lacY, and lacA gene. I hope it is clear, right? Now let's understand the regulation. How does the regulation process is happening in the presence and in the absence of lactose molecule. For understanding this, we'll consider two cases. Number one, first case, when the lactose is absent in the medium. Lactose is absent. Lactose is what? Lactose is an inducer. We have read it right now. Okay, lactose is an inducer and lactose is absent in the cell. Means what? This operon will stay in which condition? This operon will stay in off condition. And this off condition is maintained by this regulatory protein. How it is regulatory gene, sorry. How this regulatory gene maintains the off position? It synthesizes a repressor protein. Now what will happen? This repressor protein will go and bind with the operator region like this. Now when the RNA polymerase from the promoter region will start to move, what will happen? It will get obstructed here in the operator region and it will not be able to move further, and ultimately the process of transcription is stopped and the operon is in off condition. Okay, so this happens in the absence of lactose. Now moving on, the second case, that is in the presence of lactose. What happens in the presence of lactose? The regulatory protein similarly synthesizes what? A repressor molecule like this. But lactose is what? Lactose is an inducer. We have read lactose is an inducer. Lactose molecule is an inducer. What it does? It switches on the off condition of the operon. Lactose, what will lactose do? Lactose will switch on the operon. This is usually in off condition, but this will switch on. How it will switch on? The repressor protein, which is synthesized, instead of binding with the operator, it will go and bind with the lactose molecule. Now the operator molecule here, it is free. So what will happen when the RNA polymerase will start moving? It will smoothly pass over the structural gene. And as RNA polymerase reaches the structural gene, it will synthesize the mRNA. Synthesis of mRNA occurs from the structural gene, means the operon is in on condition. And ultimately, the proteins of interest or the gene products are formed. So this is the whole process of Lac Operon, the two cases of Lac Operon, that is in the presence and absence of lactose molecule we have studied. So this is all for today's video. Okay. So this is all. Thank you for listening.